Hello everyone, and welcome to another audio podcast in this series, the Pathology Storm. This is the Shientist, and I'm going to be talking to you a little bit around renal cells and COVID-19. This is a part of the Shientist chartered edition and a Storm Robotics production in collaboration with the Renal Patient Support Group, or the RPSG. In this audio podcast, I'm going to follow on from follow on from the sort of previous audio podcast, and the whole series. This whole audio podcast series relates to a peer review manuscript that has been an invited book chapter contribution to a scientific publication. In line to the previous audio podcast, I'm going to provide this perspective on renal cells and COVID-19. So, as the number of SARS-CoV-2 research increases, newer theories about the interplay between coronaviruses and kidney disease develop. Ribonucleic acid, or RNA, sequencing database assessment had demonstrated high expression of ACE2 and transmembrane protease serine 2, protease serine 2, also known as TMPR SS2, in proximal tubular cells and podocytes, which might indicate a higher affinity for SARS-CoV-2. Microscopically, the presence of ACE2 within proximal tubular cells might indicate direct interaction with coronavirus, which subsequently reflects kidney damage. However, it does not exclude the possible ischemic damage to proximal tubular cells from haemodynamic alterations, informing that the knowledge surrounding direct interactions is vague and unspecified. Following on from that, immunobiology data indicate that lower expression of ACE2 correlates with severe effects of disease owing to downregulation of ACE2, which leads to a reduced affinity effect. The severity of the chronic illness the severity of the illness increases with lower expression of ACE2 receptors, which might again contradict the correlation among a direct interaction between SARS-CoV-2 and renal cells. And so, in the peer review manuscript, there is an illustration an illustration that provides an overview of ACE2 receptor expression in human organs. I'm not going to provide a sort of an overview around the illustration or specifically, but however, I am going to highlight that the illustration does provide, or the information around the illustration provides an overview of ACE2 receptor expression in human organs, and the kidneys presents a high level of ACE2 than do the lungs to some extent, and it leads to speculation of possible direct infection of SARS or COVID-19 within kidney cells. Several investigations around the current theory of SARS and CoV-2 tropism, which leads to viral infection of proximal tubular cells. there is argument or debate that is based on the presence of viral particles within urine detected from COVID-19 patients, especially within the second and third week of infection, and when the shedding of SARS-CoV-2 occurs. However, histological examination negates the presence of active immune-mediated glomerulonephritis due to the absence of common indicators like electron-dense deposits and mild proteinuria. Moreover, the number of viral particles among several findings due to the lack of standardized sample collection and appropriate PCR assays conducted. So just to highlight that again, that moreover the number of viral particles differs among several findings due to the lack of standardized sample collection and appropriate PCR assays conducted. Data also highlight either little or no detection, and others present a high number of viral particles, which leads to inconsistent data. And so a novel infection route was identified for SARS-CoV-2, which was the spike protein attachment of transmembrane glycoprotein CD147. And again, as mentioned in a previous audio podcast, the novelty around the COVID-19 or the COVID-19 taxonomy is the surface spike proteins, which are involved in relating to causing an infection, or in relating to how individuals can get infected through from this virus because of the surface cell-like proteins. Now, in line with this, as CD147 is expressed in various cells all around the body, including blood cells and the proximal tubules of the kidney, the possible viral entry might affect the cell cycle, which affects inflammatory responses, and further development of several diseases. And due to the novelty of initial data across the literature, the idea of direct infection within renal cells is speculative. It's still speculative. And so this ends the audio podcast in this particular around this particular subject entity in this series, the pathology storm around renal cells and COVID-19. The next audio podcast will provide a more depth surrounding understanding and surrounding the analysis of COVID-19 in context of taxonomy and genera accordingly. So this ends this audio podcast. I want to say thank you for taking the time to listen. This has been the Shientist around the Shientist Chartered Edition. It's been another Storm Robustic production in collaboration with the Renal Patient Support Group. Thank you for checking this out.